Now that I've introduced you to the different types of brain cells, we're going to focus in on neurons. So this page can be titled Neuronal Anatomy, or the parts of a neuron. We can start with the soma, or the cell body, because this is the part that will be familiar to you from general biology. You can see that this is where the nucleus is. Actually, let's use a blue pen. So the soma, or the cell body, it contains the nucleus, and you may remember that the nucleus has DNA, right? And the DNA makes mRNA, which is then translated into the amino acids needed to make neurotransmitters. Neurotransmitters are the important chemicals that will go past or across the synaptic cleft to go to the next neuron. Okay, here's the amazing part. These neurotransmitters, although made in the cell body, have to be transported by cargo freight trains, basically, motor proteins, transport the neurotransmitters to the axon terminal. So they to be stored, in synaptic vesicles there. So neurotransmitters that begin their life here in the cell body are transported all the way to the end of the line at the axon terminal, and then they're stored down here in synaptic vesicles. So the synaptic vesicles store the neurotransmitters. A synapse is where an axon terminal and a dendrite of the next neuron come together. You can circle, there's one, and there's one on this picture. Maybe you can find a few more up here. Here's one, here's one, here's one, here's one. So these are axon terminals from another neuron joining up with this part of a neuron, which is called a dendrite. We'll do that in green. And then the little hatch marks that I've put on these, those are receptors that bind to neurotransmitters. One neuron might have 100,000 of these dendrites. We're using a green pen for dendrites. They receive the incoming signals from the axon terminals of other neurons. So you can understand that thousands and thousands of neurons can be all communicating to each other. And what that means, receive the incoming signals, means bind neurotransmitters that diffuse across the synaptic cleft. Okay, so let's say that this axon terminal releases neurotransmitters that bind to the dendrite, and then how does that dendrite transmit that signal to the soma? Well, what happens is that based on what the neurotransmitter was, channels in the membrane of this dendrite will open or close that will either send a negative electrical signal or a positive electrical signal toward the soma that will either say, hey, fire an action potential, or hey, don't fire an action potential. A synapse, I'm going to use a black pen for this. What we call a synapse has to include the following structures: the axon terminal, the synaptic cleft that the neurotransmitters diffuse across, and what I'll call the target membrane. Because on this page, what is the target? It's a dendrite, right? But what if this axon terminal were meeting up with the skeletal muscle, in which case that's the target membrane. So it depends on where in the body we're talking. Within the brain, it's always going to be one axon terminal to a dendrite, one neuron to another. So the axon hillock is where it's decided whether or not an action potential will be fired. Usually, that is. The problem is, look, the axon hillock is right here, the synapses are here, so that signal has to get here. And how does the axon terminal know whether or not it's been told to fire an action potential or not? Oops, purple. So this is the primary location where action potentials are fired from. Quite truthfully, there are probably a variety of places on a neuron that can start an action potential, but I'm giving you a big picture on this. If all the signals from the dendrite sum, meaning like add up, right, like math, sum high enough, then an action potential will be fired. If not, it won't be. So if the action potential is fired here, though, it's an all or nothing phenomenon, meaning that at this point, if it gets fired, and assuming that the neuron is healthy and has myelin on it, the action potential goes all the way to the end. So we call it an all or nothing phenomenon. And each action potential has the same amplitude. What I mean by that is it would be like I'm talking to you and I say, fire. That doesn't sound like I'm that excited, right? So an action potential doesn't say fire or fire, something like that, right? Instead, if there was something pretty important happening out here, and the axon hillock's receiving that information, instead of saying either fire or fire, instead what it does is it just keeps talking. Fire, 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 fire. So intensity, rather than being coded by us screaming, by an axon hillock is encoded by the frequency. Okay, so now let's give you an example of that. So let's say there was a signal coming in on this dendrite that was, -1. And let's say there was a signal coming in on this dendrite that was +1. Then you would know that these two added together, +1 plus a -1 is 0, right? So there would be no change at the axon hillock. They would cancel each other out. What if it was just this one? What if this was the only dendrite? Then it would make it unlikely that the axon hillock would fire an action potential. We would call that an inhibitory postsynaptic potential, because the potential for an action potential is not very likely now. This is inhibitory. What about this one, though? +1. That would be considered excitatory. Because it's going to tell the axon hillock, yes, you should send an action potential. Okay, what about this one? Is that one excitatory or inhibitory? Excitatory. So we call it an EPSP. See, EPSP, an EPSP. And now if you take all of this together, you've got +6 and only -1, so you still increase the likelihood of an action potential being fired here. But what about if this one were -10? Now what's happening? So now we've got -11 versus a +6 all together, because this is an IPSP, and a pretty big one. Now the axon hillock is less likely to fire an action potential. Okay, what if this one were +20? Now we've got +26 and -11, so now we are going to get a sum total that is more likely to fire an action potential. Not sure why that's blurry. There you go. Sorry, it's blurry there for a second. Okay, now this one, let's say if it's a -15. Well, now you've got -15 and -10, -25, -26, and here we've got +26. So they've now evened out again. So we're not tilting more toward the likelihood of action potential or inhibiting it. Although this still would be called an IPSP, but the total summative nature would not have an effect. And then let's go ahead and end on a positive note, +30. Now that's going to push the axon hillock over the edge and increase the likelihood that an action potential will be fired.